Assalamu alaikum students. Today we are going to study the cell mediated and human immunity. What is meant by cell mediated immunity? It means some cells mediate or release some signaling molecules which give the immune response, which start the immune response. And the human immunity mean to make immunoglobulins by plasma cells, which are known as the antibodies. So these two immunities are interlinked with each other. Let's start. For example, this is a normal cell. This is the normal cell of our body, and for example, a virus attack on this cell. This virus attack on this normal cell, entering its DNA or RNA into this targeted cell. And when the DNA or RNA of viral DNA enter into this cell, the genes present in the cell nucleus, in the DNA, are activated and they form the interferons. Interferons are basically the cytokines. There are numerous cytokines present in our bodies, just like interferons, chemokines, interleukins, tumor necrosis factor, and some others. So interferons are the cytokines. They are synthesizing after the viral infection when the DNA is entering into the cell. The interferons synthesizing and move outside the cell. They diffuse out and approaches to the neighboring cells. These are the unaffected host cells. This cell become infected host cell, and these two neighboring cells are the unaffected host cells. The interferon are actually two types. There are two types of interferons, interferon type 1 and interferon type 2. The interferon type 1 has alpha and beta interferons, whereas the type 2 interferons are the gamma interferons. The alpha interferons releases from the infected host cell and binds to the unaffected host cells. The receptors present in these cells. In our body cells, the interferon receptors are present, and these interferons are the alpha interferons. The alpha interferons, when bind to these receptors, the unaffected host cells, the DNA present in our nucleus in these cells, they synthesizing two types of enzymes. One is the protein kinase and the other is the ribonuclease. The protein kinase inhibit the protein synthesis of the virus, while as the ribonuclease inhibit the replication of the DNA or RNA of the virus. Because after the infected host cell, the cell become burst out and the viruses released from this infected host cell and attacks on the normal or unaffected host cells. And when the viruses enter their DNA or RNA into these unaffected host cells, which are the neighboring cells of this infected host cell, so these two enzymes inhibit the viral infection by the inhibition of protein synthesis of the virus, and the ribonuclease inhibit the DNA or RNA replication of the virus. Okay, these alpha interferons also stimulate the macrophage are the leukocytes. Now, before we study the next process, we should know the leukocytes. These are the hematopoietic stem cells. The hematopoietic stem cells are present in the bone marrow, and hematopoietic stem cells continuously produces or synthesizing the T cells, B cells, T cells. These are the 4 types of T cells, B cells, macrophages, neutrophils, monocytes, and natural killer cells. The hematopoietic stem cells produces the T cells, the monocytes, macrophages, neutrophils, and natural killer cells. And all the other blood cells like the erythrocytes or the platelets, they are also producing from these hematopoietic stem cells. So hematopoietic stem cells produces the monocytes and the macrophages, but macrophages are differentiated from the monocytes. Macrophages and natural killer cells and other cytotoxic T cells are patrolling in our blood when the things are quiet. But when the infection occurs, and at the site of infection, the more and more monocytes differentiated into the macrophages. And macrophages are actually the antigen presenting cells. These are the antigen presenting cells. Macrophages are actually the engulfing cells. They engulf or ingest it or consumed the microorganisms, bacteria, viruses, and other foreign objects, foreign proteins, and foreign microorganisms. So macrophages and neutrophils, they are the engulfing cells. They engulf the microorganisms actually. So, okay, the alpha interferons releases from the infected host cells, approaches to the macrophage and activate. The macrophage activated due to the interferon alpha, and they release the interleukin 1, and secondly they release interferon gamma. As we discussed, there are three types of interferons: alpha, beta and gamma. The alpha interferons released from the post-infected cells. Gamma interferon releases from the macrophage, whereas the beta interferon secretes or released from the fibroblasts, which are present in bones and muscles and other connective tissues. So macrophages are activated and they release interleukin 1. What are interleukins? Interleukins are also the cytokines, and when they release from the leukocytes or white blood cells, they are known as the lymphokines. These are at least 35 types of interleukins. So interleukins are also the cytokines. They are signaling molecules. They respond and they are activated by these immune cells activated by these cytokines like interferon and interleukins. So interleukins are of 35 types. So macrophages releases interleukin 1, and this interleukin 1 is activated the T cells. So we discuss the T cells. The T cells are actually produces from the hematopoietic cells, and they originate from the hematopoietic cells. Once they goes to the thymus, thymus is a gray gland, gray small gland just above our heart. And when the T cells approaches or enter into the thymus, they reside here and become mature. And after maturation, the thymus produces 4 types of T cells. One is the inducer T cell, helper T cell, cytotoxic T cell, and suppressor T cell. The T cells present the antigen recognizing receptors. The antigen recognizing receptors like CD3. The CD3 is present in all 4 types of T cells, while the CD4 present only in inducer T cell and helper T cell, while as CD8 recognizing antigen receptor present only in cytotoxic T cell and suppressor T cell. All the T cells have different functions in immunity. The macrophage, when the macrophage activated, they produces interleukin 1. The interleukin 1 activated the helper T cell, which is most important T cell in immunity. And when the helper T cell activated, it produces interleukin 2. And when it produces interleukin 2, interleukin 2 also activates the thymus and produces more and more T cells or helper T cells. This mode of action of interleukin 2 is known as the autocrine effect. autocrine effect, because the cytokines has three mode of actions. One is the autocrine, the second one is the paracrine, the third is the endocrine. Autocrine mean a cell mediator or interleukin secreted from a cell and effect on its own. Paracrine mean when the mediator release from a cell and effect on the neighboring cells or other cells, which is the mode of action is known as the paracrine. So more and more T cells are producing because infection is there, and the immune system is trigger on. Also interleukin helper T cell also produces interleukin 2, interleukin 4 and interleukin 5, and these interleukins or activate the B cells. B cell has many receptors, at least 1 lakh receptors are present on B cells, and after this activation, because interleukin 2, 4, 5 activate the B cell, and the B cell when activated they differentiate into the plasma cell, and plasma cell produces the immunoglobulins, which are known as the antibodies against the antigens. The helper T cells also activate the cytotoxic T cells and also activate the inducer T cells. The cytotoxic T cells releases some poisoning substances, some poison-like substances or some chemicals which puncture or lyses the infected cell and puncture this cell to become a hole, and the water diffuses into this cell, infected cell, by osmosis. Finally the cell becomes burst out, and the macrophages and neutrophils, they engulf, ingest and consume. And after consumption and after they consume, the macrophages and neutrophils, these foreign proteins which are known as the antigens, they display on their surface like a victory banner. So these are the antigens present, the black color, these are the antigens present on the surface of the macrophage, because the macrophage is the antigen presenting cell, and these are the receptors MHC2 and the MHC1. What are these? MHC mean major histocompatibility complex protein. These are actually the self marker, and MHC1 major histocompatibility complex protein are present in all nucleated cells of our body. Just like this, MHC1. But MHC2 are present only on the antigen presenting cells like the macrophages, neutrophils and some other cells. So the cell, the body cell display the antigens. Antigen mean any foreign body, any foreign protein, which is known as the antigen, which is the protein which is not self, which is come from the outside, the microbial protein actually, and this which are presented on the surface of the cell, which is known as the antigen, but present in and loading in MHC1 marker. MHC1 is the marker, and the antigen is presented in MHC1 by infected host cell, while as the macrophage presenting the antigen in MHC2, because the infected host cell nothing has MHC2. MHC2 only present in the macrophage. So these are the antigens presented by the infected host cell and the macrophage. And now the helper T cell and cytotoxic T cells and also the B cells recognizing these antigens and attract towards these antigens, and for this purpose the helper T cell has the CD4 marker, which is not self. Not self mean this is the recognizing receptor, recognizing marker present on the helper T cell, and the cytotoxic T cell has CD8 marker, which is also recognizing. But the helper T cell recognized the MHC2, and after the loading of the antigens, if the antigens are not there, if the infection is not there, the marker is present, antigen is not present due to the lack of infection. So CD4 is not recognizing this marker, MHC2. CD4, the helper T cell, only recognizing MHC2 when the MHC2 loaded the antigens. And the cytotoxic T cells, which has the marker CD4, recognizing the infected host cell only when the antigen is present or antigen is loaded in MHC1. So CD8 recognizing only MHC1, the helper T cell recognizing MHC2. And after this recognition, a complex is formed. Here, the antigen presenting cell like macrophage, which has MHC2 receptors, MHC2 marker, red line, red area. This is the marked by red marker. So MHC2, and this is the antigen. And the helper T cell, HTC mean helper T cell, recognize the MHC2 because the antigen is present and attach to the macrophage. Whereas the B cell, the B cell recognize also the MHC2 when the antigen is present on the surface of the macrophage, attached with the macrophage. And why we are called as the helper T cell? Because they help. These T cells help the B cells. Why this? How this help is possible? This help possible by the secretion or by the release of lymphokines. What are lymphokines? Basically they are cytokines. And these cytokines are different types. One of the cytokines are the interleukins, interleukins 2, interleukin 4, interleukin 5. As we discussed earlier, the helper T cell secreted interleukin 2, interleukin 4, interleukin 5. So, helper T cell secretes interleukin 2, interleukin 4, interleukin 5 to the B cells. The B cell activated and produce and differentiate the plasma cell. And plasma cell produces or manufacture or releases the antibodies, which are the immunoglobulins, against these antigens which are present by the antigen presenting cell, by the macrophage. So this is the function of the helper T cell and B cell. What is the inducer cells? The function of the inducer cells. Inducer cells has the marker of CD4, also the CD3, because the CD3 marker is present in inducer T cell, helper T cell, cytotoxic T cell and suppressor T cells. In all T cells, the CD3 is present. And what is the function of the CD3? The function of the CD3 is very simple. When the cells are attacked with the antigen presenting cell, the signals pass from the CD3 to the inner cells. That cell is occupied. This is the signaling informations. The informations passed from the CD3, which is present on the helper T cell, actually, to inside the macrophage. That cell is occupied. And what is the function of the inducer T cell? The inducer T cells are helped to manufacturing or producing more and more T cells from the hematopoietic stem cells. They induce the informations that infection is there. And after this infection, inducer T cells passes these informations to the hematopoietic stem cells. That hematopoietic stem cells in response, they produces more and more T cells. And what is the function of the suppressor T cells? Suppressor T cells actually suppress the cytotoxic T cells, or the function of the cytotoxic T cells, which shut down the immunity, and also they suppress the B cells which shut down the immune response, and this is the negative feedback actually. So negative feedback is the responsibility of suppressive T cell is the negative feedback. What is the function of the cytotoxic T cells? Cytotoxic T cells doesn't attack directly on the function of the cytotoxic T cell is to kill and mark for destruction the infected host cell, because cytotoxic T cell recognizes the MHC1 loaded with antigens, and for the recognition it has the CD8 receptors present on cytotoxic T cell. So CD8 recognizes the MHC1 marker loaded with antigen, and this happened this is possible because due to the infection. So cytotoxic T cell mark for destruction the MHC1, it lyses and create a hole, the cell, the water enter into the cell become burst out, and when the cell become burst out, the debris of the cells ingested by the macrophage and neutrophils and clean the way. Here we discuss the some role of the B cells, where the B cells differentiated into the plasma cells, and the plasma cells produces the immunoglobulins. Some B cells remains as the memory cells, and when after healing the patient after this infection, when the second attack of this type of organism, the B cells remains as the memory cells, and B cells immediately differentiate into the plasma cell and produces the immunoglobulins. The role of the interleukin 1 is also to increase the temperature, This is the interleukin 1, because interleukin 1 boost or stimulate the hypothalamus. The hypothalamus is the part of the forebrain, and the hypothalamus is a thermoregulator, and after this stimulation by the interleukin 1, the temperature rises, and the hypothalamus sending the signals to our muscles to contract more and more, to the blood vessels where they shrink or minimize the heat loss from the skin. As a result, the shivering starts and the temperature rises. The rising of the temperature is also the part of the immune response, and this rising of temperature is also the part of the innate immunity as well as the part of the cell-mediated immunity. The final thing is the natural killer cells. They are producing from the hematopoietic stem cells. These are also the leukocytes, and the natural killer cells does not kill the microorganisms directly, but they mark for destruction, because natural killer cells pulse the infected cell by lysing, and they do like the cytotoxic T cells, and the cell become burst out. So this is all about the cell-mediated and humoral immunity, and the role of the T cells, especially the helper T cells, macrophages, and the cytokines, the interferons, interferon alpha, interferon gamma, and interleukins are secreted from these cells. They act on these cells to activate them, and these cells are show the immune response and complete the cell-mediated and humoral immunity. So this is all about the immunity, and request for subscribing my channel, like and share, because these are the energy dose for our, and I hope I will prepare more and more knowledgeable videos for you in future very soon. Thank you very much.